Assalamualaikum warahmatullahi wabarakatuh. Saya Riska Novralani, mahasiswi STIFI BHAKTI PERTIWI Palembang semester 6. Jangan lupa nonton sampai selesai. Nah, di sini saya akan menjelaskan apa itu analgetik, apa itu Antiparkinson, hipnotik-sedatif itu apa, dan contoh-contohnya itu gimana. Kita simak video berikut yuk.
In this lecture, we are going to cover the pharmacology of opioids, so let's get right into it. Opioids, sometimes called narcotics, are a group of drugs that act on the central nervous system to produce morphine-like effects such as pain relief and euphoria. Now, in order to gain better understanding of their mechanism of action, first we need to talk about the transmission of pain. So pain begins at the nociceptors, which are simply the branching ends of sensory neurons found within the peripheral nervous system. These high-threshold primary sensory neurons respond to damage to the body by transmitting the painful stimulus to the second-order neurons in the dorsal horn of the spinal cord. From there, the signal is carried through the spinothalamic tract to the thalamus, and then to the somatosensory cortex, where pain is perceived. Now, on a microscopic level, the pain signal takes the form of a series of action potentials that fire repeatedly depending on the intensity of pain. To enhance movement across the synaptic cleft, transmitter chemicals are released from the presynaptic neurons, including glutamate, substance P, and calcitonin gene-related peptide, CGRP for short. Glutamate is one of the most important neurotransmitters for pain, and can activate both NMDA and AMPA receptors, which permit influx of positively charged calcium and sodium ions respectively. As you may recall, the flow of positively charged ions into the neuron makes the neuron more likely to fire. In this way, glutamate excites the second-order neurons in the dorsal horn, which leads to propagation of a sharp localized pain signal. Substance P, on the other hand, binds to the neurokinin-1 receptor, NK1 for short, which leads to intracellular signaling that involves activation of arachidonic acid pathways, nitric oxide synthesis, and activation of NMDA receptors. NMDA receptors are activated when substance P attaches to NK1 receptors and then gets incorporated into the cell, activating protein kinase C. This action removes the magnesium that under normal conditions is blocking NMDA receptor. This in turn allows glutamate to attach to the NMDA receptor and thus permit the inflow of calcium ions, ultimately causing the pain signal to increase and fire more frequently. Lastly, the released CGRP binds to its receptor on second-order neurons, leading to changes in receptor expression and function, and thereby altered neuronal activity. This in turn contributes to the so-called central sensitization that is characterized by a lower threshold for evoking action potentials. Now, fortunately for us, our bodies can cope with a certain amount of pain by releasing so-called endogenous opioids. There are three major families of endogenous opioids: the enkephalins, dynorphins, and endorphins. Endogenous opioids exert their effects by binding to opioid receptors, which are abundantly present in the central and peripheral nervous systems. There are three major types of opioid receptors, that is mu, delta, and kappa. In general, all three receptors differ in their cellular distribution, their relative affinity for various opioid ligands, and their contribution to specific opioid effects. All opioid receptors are 7 transmembrane spanning proteins that couple to inhibitory G proteins, and they are all present in high concentrations in the dorsal horn of the spinal cord. Activation of these receptors by an agonist, such as the endogenous mu opioid peptide endorphin, causes closing of the voltage-gated calcium channels on the presynaptic nerve terminals, which in turn decreases the release of neurotransmitters such as glutamate, substance P, and calcitonin gene-related peptide. In addition to that, activation of opioid receptors leads to opening of potassium channels, allowing efflux of potassium ions, which in turn results in hyperpolarization, rendering neurons less sensitive to excitatory inputs. Now, the majority of currently available opioid analgesics act primarily at the mu opioid receptors, essentially mimicking the effects of endogenous opioid peptides. However, while naturally derived opioids can only reach a certain potency, the synthetically produced opioids are refined and processed to be much more powerful. The examples of synthetic opioid agonists are fentanyl, hydrocodone, hydromorphone, methadone, meperidine, oxycodone, and oxymorphone. As a side note here, it's important to note that methadone is not only a potent mu receptor agonist, but also a potent antagonist of the NMDA receptor, as well as norepinephrine and serotonin reuptake inhibitor. These properties make methadone useful for treatment of both nociceptive and neuropathic pain. Now, in addition to producing analgesia, activation of the opioid receptors in other parts of the body can bring about many side effects. For example, all opioids produce some degree of nausea, which is due to direct stimulation of the chemoreceptor trigger zone in the medulla. All opioid receptor agonists also produce a dose-dependent respiratory depression. Opioids primarily cause respiratory depression by reducing brainstem respiratory center responsiveness to carbon dioxide. They also depress the respiratory centers in the pons and medulla, which are involved in regulating respiratory rhythmicity. In addition to that, opioids produce an antitussive effect by depressing the cough center in the medulla. Opioids are known to be associated with suppression of the immune system, as opioid receptors are involved with regulation of immunity. Morphine as well as meperidine may provoke release of histamine, which plays a major role in producing hypotension. Furthermore, when given by injection, morphine and meperidine can cause dilation of cutaneous blood vessels, which results in the flushing of skin of the face, neck, and upper thorax. Meperidine in particular produces tachycardia due to its structural similarity to atropine. Other opioids generally produce a dose-dependent bradycardia by increasing the centrally mediated vagal stimulation. All opioids can cause itching via central action on proprioceptive neural circuits. Opioids also decrease gastric motility and prolong gastric emptying time, which may cause constipation. Likewise, opioids depress renal function and produce antidiuretic effects. They also increase sphincter tone and thus may cause urinary retention. Now, the biggest problem with opioids is that they have the potential to cause addiction by causing both physical and psychological dependence. The euphoric effect appears to involve GABA inhibitory interneurons of the ventral tegmental area of the brain. Normally, GABA reduces the amount of dopamine released in the nucleus accumbens, which is a brain structure that is part of our pleasure and reward system. However, when opioids attach to and activate the mu receptors in that area, the release of GABA becomes suppressed. This in turn increases dopamine activity and thereby increases the amount of pleasure felt. Now, on the other hand, prolonged regular use of opioids leads to desensitization of receptor signaling and downregulation of the receptors and thus a decrease in sensitivity to the effects of opioids. As a result, when regular opioid use is reduced or suddenly stopped, the lack of receptor activity is manifested as withdrawal symptoms. These symptoms generally are opposite to the pharmacological effects of the opioid drugs. So now, rather than causing constipation and slowing respiration, the brainstem triggers diarrhea and elevates blood pressure. Instead of triggering happiness, the nucleus accumbens and amygdala reinforce feelings of dysphoria and anxiety. All of this negativity feeds into the prefrontal cortex, further pushing a desire for opioids. Now, before we end, I wanted to briefly discuss a couple more agents that interact with opioid receptors, but in a different way than the agents that we discussed so far. The first one is a partial mu receptor agonist called buprenorphine. So while a full opioid agonist binds to the mu receptor, activates it by changing its shape and thus induces a full receptor response, a partial agonist binds to the receptor and activates it with a smaller shape change, which leads to only a partial receptor response. In other words, the effects of partial agonists increase only until they reach a plateau. Like all opioids, buprenorphine can cause respiratory depression and euphoria, but its maximal effects are much smaller than those of full agonists. The benefits of this are lowered risk of abuse, addiction, and side effects. One last thing to keep in mind is that buprenorphine is also an antagonist at the delta and kappa receptors, and because of that, it is referred to as mixed agonist-antagonist. However, the contributions of these actions to its analgesic profile are currently unclear. Now let's move on to our last agent, that is naloxone. So naloxone is an opioid antagonist that can be used to block or reverse the effects of opioid drugs. Naloxone works by knocking off the opioids attached to the receptors in the brain, thereby temporarily stopping the opioid effect. This is possible because naloxone has a stronger affinity for opioid receptors, and thus is able to kick the opioids out and block them from attaching again. So during an emergency situation, when a person's breathing has slowed down or stopped due to an opioid overdose, naloxone can quickly restore normal breathing and save the life. And with that, I wanted to thank you for watching. I hope you found this video useful, and as always, stay tuned for more. In this lecture, we're going to cover the pharmacology of anxiolytic and hypnotic drugs, so let's get right into it. The primary use of anxiolytic and hypnotic drugs is to treat anxiety and insomnia. Anxiety is generally defined as an emotional state characterized by excessive feelings of worry, fear, and nervousness, while insomnia is generally defined as the inability to initiate, maintain, or achieve restful sleep. Now, in order to gain better understanding of how anxiolytic and hypnotic drugs work, first we need to discuss the role of GABA in the central nervous system. So, gamma-aminobutyric acid, or GABA for short, is a major inhibitory neurotransmitter. What that means is that when GABA reaches and binds to a specific receptor located on a neuron, it reduces the activity level of that neuron and makes it less likely to fire its action potential. Now, GABA is recognized by both ligand-gated and G-protein-linked GABA receptors. In this lecture, we are going to focus on the ligand-gated GABA receptor called GABA-A, as this is the main target of many anxiolytic and hypnotic agents. Now, the GABA-A receptor is made up of 5 subunit proteins arranged to form a central pore. These subunits have been labeled with Greek letters such as alpha, beta, gamma, and delta. The subunit composition of GABA-A receptors in the brain most frequently consists of 2 alpha subunits, 2 beta subunits, and 1 gamma or delta subunit. Furthermore, for each subunit, many other subtypes exist. The function of each GABA-A receptor depends on which subunit it contains. For example, GABA-A receptors containing alpha-1 subunit appear to be involved in sleep, while those that contain alpha-2 or alpha-3 appear to be involved in anxiety. Now, when 2 GABA molecules bind to the GABA-A receptor, conformational change is induced, which results in opening of the central pore that allows negatively charged chloride ions to rush into the cell. This in turn increases the negative charge inside the postsynaptic cell, which makes more difficult for an excitatory inputs to cause an action potential. Now, GABA-A receptors are present in high concentration in the limbic system. One part of the limbic system is the amygdala, which contains many neurons that use the neurotransmitter GABA to control feelings of fear and anxiety. In times of stress, excitatory neurons in the amygdala fire rapidly, sending excitatory signals to other areas of the brain. It is this type of firing that leads to feeling of panic or fear. The inhibitory interneurons in the amygdala regulate these emotions by releasing GABA. The release of GABA and its binding to postsynaptic receptors such as GABA-A inhibits the excitatory signals that result in feelings of anxiety and fear. GABA thus has a calming, tranquilizing effect on our emotions and prevents us from becoming overwhelmed in stressful situations. Now, in addition to a distinct binding site for the neurotransmitter GABA, the GABA-A receptor has also a site at which anxiolytic and hypnotic drugs bind. These drugs can be divided into 3 groups, that is benzodiazepines, barbiturates, and non-benzodiazepine hypnotics. Let's now discuss these one by one in detail, starting with benzodiazepines. So benzodiazepines get their name from their chemical structure, which includes a benzene ring fused to a diazepine ring. Their binding site appears to be the region between the alpha and gamma subunit on the GABA-A receptor. It's important to remember that benzodiazepines do not bind to the GABA recognition site located at the junction of the alpha and beta subunits, thereby they don't induce opening of the channel as directly as GABA. Instead, the binding of a benzodiazepine to its receptor site increases the affinity of GABA for the GABA binding site. This leads to increased frequency of the channel opening, and in turn increased influx of chloride ions at the postsynaptic cell, which ultimately makes it difficult for any excitatory inputs to cause an action potential. So when the excitatory signals caused by fear or stress are inhibited in this way, the result is decreased anxiety and greater feeling of calm. In addition to that, benzodiazepines can also produce mild muscle relaxation as a result of their action on GABA receptors in the spinal cord, cerebellum, and brainstem. The examples of benzodiazepines are alprazolam, chlordiazepoxide, clonazepam, diazepam, lorazepam, and temazepam. When it comes to side effects of benzodiazepines, the most common ones are related to their sedating and muscle relaxing action. They include drowsiness, dizziness, decreased alertness and concentration, as well as impairment of motor coordination. Now let's move on to the next group, that is barbiturates. So barbiturates bind to GABA A receptor at the alpha or beta subunit, which is different from the GABA binding site and also different from the benzodiazepine binding site. Now, just like benzodiazepines, barbiturates enhance the effect of GABA at the GABA A receptor. However, instead of increasing the ion channel opening frequency, they increase the ion channel opening duration. In other words, unlike benzodiazepines, barbiturates hold the ion channel open continuously for long periods of time. In addition to that, barbiturates can decrease the activity of excitatory neurotransmitters such as acetylcholine and glutamate, and at higher doses, they can also mimic GABA and directly activate GABA receptors, thus leading to profound central nervous system depression. This is why barbiturates have a much lower therapeutic index than benzodiazepines, meaning the toxic dose is not much higher than the therapeutic dose. The examples of barbiturates include pentobarbital, phenobarbital, and secobarbital. When it comes to side effects, barbiturates most often cause dizziness, lightheadedness, sedation, as well as memory and attention impairments. Furthermore, because of their narrow therapeutic index, it's important to remember that even small barbiturate overdose can result in coma or death due to respiratory depression. Now let's move on to our last group of drugs, that is non-benzodiazepine hypnotics. As I mentioned at the beginning of this lecture, GABA A receptors in different areas of the central nervous system consist of various combinations of the subunits. The specific subunit that appears to mediate sedation is the alpha-1 isoform. So as their name suggests, non-benzodiazepine hypnotics are structurally different from the benzodiazepines, and as you may already guess, they selectively bind to the alpha-1 subunit of the GABA A receptor, thus triggering chloride channel opening with resulting membrane hyperpolarization. Because alpha-1 containing GABA A receptors are highly expressed in wake-promoting regions of the brain, the preferential binding of non-benzodiazepine hypnotics to these receptors results in sleepiness but not anti-anxiety effect. The examples of non-benzodiazepine hypnotics are zolpidem, zaleplon, and ezopiclone. When it comes to side effects, non-benzodiazepine hypnotics can cause cognitive impairments, including memory loss, daytime sedation, as well as impairment of motor function. And with that, I wanted to thank you for watching. I hope you enjoyed this video, and as always, stay tuned for more. In this lecture, we are going to cover the pharmacology of drugs for Parkinson's disease. So let's get right into it. Parkinson's disease is a neurological disorder that results in a progressive loss of coordination and movement. Now, the neurons responsible for coordinating movement are located in a part of the brain called striatum, which receives information from two major sources, the neocortex and a region known as the substantia nigra. The cortex relays sensory information as well as plans for future action, while the substantia nigra sends dopamine that helps to coordinate all of the inputs. Now, Parkinson's disease develops when the neurons connecting the substantia nigra to the striatum progressively degenerate. Since dopaminergic neurons that originate in the substantia nigra normally exert inhibitory effects on GABA neurons located in the striatum, too little dopamine results in more GABA causing increased inhibition of the thalamus, as well as reduced excitatory input to the motor cortex. In addition to that, these same dopaminergic neurons also exert inhibitory effects on the excitatory cholinergic neurons in the striatum. And again, without sufficient levels of dopamine, the production of acetylcholine is increased, which triggers a chain of abnormal signaling leading to impaired mobility. Ultimately, the imbalance between inhibitory and excitatory activities leads to the manifestation of typical clinical symptoms that include resting tremor, rigidity, postural instability, and slowed movement. The pharmacological therapy for Parkinson's disease is aimed at replenishing dopamine levels, mimicking dopamine's action, or antagonizing the excitatory effects of cholinergic neurons. Now, in order to get a better understanding of the pharmacology of anti-Parkinson agents, first we need to take a closer look at the dopamine-producing neuron. So here, inside this dopaminergic neuron, dopamine is synthesized in a two-step process, starting with the amino acid tyrosine. First, with the help of enzyme tyrosine hydroxylase, tyrosine gets converted to L-dopa, also known as levodopa. In the second step, the L-dopa formed by tyrosine hydroxylation is quickly decarboxylated by another enzyme called aromatic L-amino acid decarboxylase to the neurotransmitter dopamine. Dopamine is then loaded into synaptic vesicles and released by physiological stimuli into the extracellular space where it can bind to dopamine receptors that are expressed on the postsynaptic neuron. Finally, excess dopamine in the synapse is reuptaken back into the neuron or into glial cells where it gets metabolized by monoamine oxidase, abbreviated MAO, and catechol O-methyltransferase, abbreviated COMT. It's important to note that while the MAO enzyme exists in two forms, known as type A and type B, the type that is predominantly found in the glial cells is the MAO type B. Now let's move on to discussing drugs used in treatment of Parkinson's disease, starting with one of the most commonly used drugs, that is levodopa. But first things first, you may wonder, well, why would you use the precursor of dopamine instead of dopamine itself? And the answer is, it's because of the blood-brain barrier. Blood-brain barrier is a tightly packed layer of endothelial cells that restricts free access of molecules between the blood and the brain. Dopamine happens to be one of those molecules that cannot freely pass through this barrier. However, levodopa can, with a little bit of help. One of the biggest problems levodopa faces on its own is peripheral metabolism. There are two major enzymes in periphery which cause breakdown of levodopa before it can reach the brain. That is peripheral dopa decarboxylase, which converts levodopa to dopamine, and catechol O-methyltransferase, which converts levodopa to 3-O-methyl-dopa. Because of this, levodopa must be administered with another agent, called carbidopa, which inhibits dopamine decarboxylase and thus reduces metabolism of levodopa in the periphery. Another agent that is used in combination with levodopa and carbidopa is entacapone, which inhibits peripheral COMT, and thus, just like carbidopa, it prolongs the time that levodopa is available to the brain. Now, levodopa is carried across blood-brain barrier by amino acid transporter. Once inside the brain, levodopa is efficiently converted to dopamine, thus supplementing the depleted dopamine levels in the midbrain. However, let's not forget that dopamine is also susceptible to breakdown by COMT as well as MAOB, which convert dopamine to 3-methoxytyramine and 3,4-dihydroxyphenylacetic acid respectively. So here another useful drugs come into play, namely selegiline and resegiline, which selectively inhibit MAOB, and tolcapone, which inhibits COMT. As a side note here, in comparison to entacapone, tolcapone can better penetrate the blood-brain barrier and thus can act both in the central nervous system and in the periphery. So again, as you can see, by decreasing the metabolism of dopamine, these drugs help to increase dopamine levels in the brain. Unfortunately, because Parkinson's is a progressive disease, with time the number of dopamine-producing neurons decreases, and fewer cells are capable of making dopamine. Taking that into consideration, some drugs have been developed to mimic dopamine and directly stimulate dopamine receptors in the brain. Drugs that belong to this class include bromocriptine, ropinirole, pramipexole, rotigotine, and apomorphine. Now, as I mentioned at the beginning of this lecture, in Parkinson's disease, dopamine depletion leads to increased acetylcholine release, which then activates muscarinic receptors located on the neurons responsible for smooth motor control. The overstimulation of these neurons by acetylcholine then causes tremors and rigidity. This is where antimuscarinic agents come into play by blocking the muscarinic acetylcholine receptors and cholinergic nerve activity. As a result, these anticholinergic agents restore the balance between acetylcholine and dopamine, which may improve the symptoms of Parkinson's disease. Drugs that belong to this class include benztropine, bipyridine, procyclidine, and trihexyphenidyl. Now, before we move on, I wanted to briefly mention here one more drug used in the treatment of Parkinson's disease, that is amantadine. Amantadine doesn't exactly fit into any of the classes that we discussed so far. Its mechanism of action is poorly understood. However, some of the speculated ones are that it prevents dopamine reuptake, facilitates presynaptic dopamine release, and blocks glutamate NMDA receptors. Now, when it comes to side effects, levodopa in combination with carbidopa may cause nausea, loss of appetite, hypotension, mental disturbances, and discoloration of urine, sweat, or saliva. Selegiline and resegiline may cause nausea, insomnia, dyskinesia, and visual hallucinations. Entacapone and tolcapone may cause discoloration of urine, sweat, or saliva, and diarrhea, which can get severe, particularly with the use of tolcapone. In addition to that, tolcapone has been associated with liver toxicity. All dopamine agonists may cause nausea, orthostatic hypotension, mental disturbances, and daytime sleepiness. In addition to that, bromocriptine has been associated with pulmonary and cardiac fibrosis. Lastly, drugs that block muscarinic receptors generally produce anticholinergic side effects, such as constipation, urinary retention, dry mouth, and blurred vision. And with that, I wanted to thank you for watching. I hope you found this video useful, and as always, stay tuned for more.